Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. First, we'd like to thank our Dr. GPCR Ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Last month, we launched the Dr. GPCR Symposia event series. Our first symposium was on March 24th, 2023, and it was on challenging GPCRs. It was a day filled with amazing talks, great discussions, and we hope the beginning of new collaborations. In case you missed it, you can watch the talks in the Dr. GPCR ecosystem with your premium membership. Also, mark your calendars for the next symposium coming up on May 19th, during which we will hear from speakers on GPCR activation and signaling. At all our symposia events, including the one coming in May, all trainees are welcome to present a poster. There won't be any poster selections, and everyone is welcome to join us for the poster/networking time. For more information and an updated schedule can be found in the ecosystem. The easiest way to get to it is to use the links in the footer and look for Dr. GPCR Symposia. You can join us live by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. To navigate the ecosystem again, please use the direct links in the footer. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have the pleasure of having with me Aditya Kumar. Happy to have you on. It's so nice to finally get to chat with you. I know we've interacted a little bit at some of the Dr. GPCR events, but welcome to the podcast.
Thank you so much, Emin. It's such a pleasure. And I am a regular listener of the podcast, I would say, and a more or less regular attendee of various events that you do. So really happy to be here.
Thank you. Thank you. I sometimes see your name pop up on Zoom at different events. And I remember you asking very sharp questions at some of these virtual cafes, which we don't host anymore because
That's sad.
yes and no. We realized that the attendance wasn't what we wanted it to be. So that's why we decided to kind of revamp and switch to the one-day symposium throughout the year. And also there will be an opportunity. I'm thinking of having kind of a journal club type of event because it's been difficult to get trainees to come either on the podcast or to come and present. So I'm trying to. I'm sorry.
Forward.
Yes, absolutely.
Yep.
I think that's going to be great, and I think it removes the pressure as well when it's a journal club. Most of the time it's trainees who attend, so I think from a presenter's perspective it's less of a stressful event. All right, so let's start at the beginning. You are in Ann Arbor, Michigan.
Yes.
And it's very cold. It's just as cold here in Boston, I think.
Yes.
So why don't you introduce yourself a little bit?
Okay, so I was born and raised and completely educated in India. For those who understand India slightly better, I introduce myself as genetically a South Indian and epigenetically a North Indian because I was born in the South and we are natively from the South of India, but I mostly grew up in the North. Education-wise, as an undergrad, I did a bachelor's of technology, which is curiously an engineering degree for those outside India, in biotechnology, which is a 4-year undergrad course. And then right after that, I went on to interview for PhD positions in India. Interestingly, I never thought I would be a scientist. I listened to a lot of people, scientists, introducing themselves as saying that, oh, when I was 6, I wanted to be a scientist. That is not the case with me at all. Science grew on me, and I'm so happy that that is the case. I didn't go into this BTech degree thinking that I would, at the end, apply to PhD positions and do professional science. But it just so happened that through time, I kind of enjoyed the whole process of science. I mean, doing science professionally, as I did a few projects here and there, and then the whole part where I went to different places to interview for PhD positions, and I just like saw labs and these famous people you hear about, interviewing with them, sitting across a table or at a whiteboard, trying to answer questions for 40 minutes. That whole experience was very exciting for me, and I decided that this is what I want to do. I ended up eventually at Hyderabad at the Center for Cellular and Molecular Biology, which is a premier life science, modern contemporary life science research institute in India. And after a few rotations in different labs, I ended up joining Professor Amit Chattopadhyay, who essentially is the lab works on membrane biology, at the interface of membrane biology and receptors. We mostly identified as membrane biologists slash biophysicists, I would say, in Amit's lab. That is where I kind of stumbled across my first lab, I would say, is membranes. And GPCRs were like a thing, I would say, a tool to study lipid protein interactions in membranes and different kinds of membranes. We studied right from natural membranes from native sources, like hippocampus from a buffalo brain, for example, and then right to cellular systems. And then also there was an interesting aspect where we were looking at the role of membranes in lipid protein, in entry of pathogens into cells. And a major focus of the lab then was also on fluorescence spectroscopy of model membrane. So it was a very, wide variety of stuff happening in the lab, and very exciting to have like colleagues with different backgrounds. There were people with chemistry degrees and physics degrees, and like people trained in core biology and so on. So that was an exciting phase, and that is when I got introduced to GPCRs. And I know that's going to be your next question, so
Yes,
I'm already
yes.
answering it.
I did.
So as far as GPCRs go, Amit had been studying GPCRs and the role of lipids and how lipids contribute to the function and organization and dynamics of GPCRs in various kinds of systems. He had been doing that, and when somebody joined the lab, they had this option of trying to like pick from all of these avenues and what do you want to do, what interests you, and so on. And the thing there was also that you can like jump on multiple boats and see what fits you best, and do whatever you like the most, and so on. So when I started in Amit's lab, I took up a completely different project nobody had really done before. And I was given the task of trying to explore the endocytosis and trafficking of the receptor of interest in the lab, it's been a receptor of interest for a long time now. It's the serotonin 1A receptor. It's a class A GPCR. So I was told by Amit that, look, this is the part where that we haven't done at all. They had looked at cholesterol, interaction of cholesterol with receptors, and how if you like, modulate lipid levels in membranes, how that would affect receptor function and dynamics and organization using several tools, including like core pharmacology, like radioligand binding assays to G protein coupling assays to doing FRET experiments and FRAP experiments to look at dynamics and oligomerization in live cells. And we collaborated heavily to do molecular dynamic simulations on these as well, to study all of these aspects. But endocytosis and trafficking was not something that we had done before, and I had the challenge of initiating that project. So and it was both exciting and challenging, I would say, and took some time to get going. But yeah, that is something that I established. And um, then I looked at the role of membrane lipids in the organization of receptors in cells. Sorry, membrane cholesterol in
Yeah.
trafficking and
Yeah.
endocytosis of these receptors in cells. Um, yeah, and I also had a side project about how this pathogen called Leishmania donovani. It's an interesting pathogen. It causes leishmaniasis in a lot of tropical areas um, in India, in Africa, in
Yeah.
Brazil, and so on, and how this pathogen actually enters into cells. So that was another project that I did. So after all of this, I ended up getting my PhD, and I moved on to the US, where I am now at the University of Michigan at Ann Arbor, in the medical school, in the Department of Pharmacology. I'm with Manoj Putanvedu, um, who is famous for looking at the trafficking and signaling and the interplay between trafficking and signaling of GPCRs inside cells. And yeah, my saga continues here.
That's amazing. And I have a couple of follow up, and I want to take you back a little bit for those questions, but just putting it out there, if Manoj is listening to this episode, he said, yes, I'm coming to the podcast. And I think we're almost at the 12-month mark where, and I know he's busy because like, yeah, I have to finish this and then I'm coming. So if you're listening, Manoj, you're still welcome on the podcast. It's an open
I will
invitation.
I will also put in a word for you. And I
Thank you.
know he has been, but it's just
I know.
there's so many
I
things to
think
do, I guess.
COVID just messed up a lot of things. And it's funny because a lot of our previous PI guests were invited to the podcast, and then they were like, yeah, I'm coming. And then I ended up inviting someone from their lab, and they're like, how come you were first on? And that
Nice.
gave the little push.
Nice.
And
Okay.
it wasn't intentional at all. We're always
Hmm.
looking for guests, and I'm putting it out there. We're always looking for guests as long as you're working in the GPCR field. Even if you're a trainee and you feel like you don't have that 25-year experience, you are an expert at your own project.
Right.
You have an experience that is worth sharing because there are people who are junior compared to you who can use your hearing more about your experience. So that was my spiel to get trainees on the podcast. So
Awesome.
if we haven't reached out and you want to come on the podcast, just email us. I think even in the ecosystem, we do have a little form that you can fill out to join us on the podcast. I don't think anyone has ever filled out a form. So I don't know. I think we're getting there. And thank you again for coming. All right, so let's take you back. You said that science kind of grew on you. And I'm interested in hearing more about your first encounter with science, where you were like, I actually might like this. Do you remember that moment?
Mm, I would say I during my I mean, actually I was always interested in science. I mean, as a kid going to school, I would always be among the first people to uh, attend science exhibitions or even organize them, like have stalls there and so on. So and participate in quizzes. I mean, quizzing is something that is very close to my heart. So I was always part of like science
Yeah.
quizzes and general
Mhm.
quizzes and so on.
Yeah.
So I always enjoyed science, but the idea of like doing science professionally, like on the bench,
Yeah.
I would say occurred to me as I was doing a few projects during my undergrad.
Yeah.
I remember having traveled to Bangalore with a bunch of my friends to spend a couple of months doing a computational project on drug design and stuff like that. And
Wow.
it was, I mean, in retrospect, pretty silly, I would say. I mean, it was like looking at a few compounds, trying to dock them and trying to see
Mhm.
which fits best, and energy minimization of molecule and
Yeah.
so on. I don't even remember most of it right now, but that was pretty exciting. But then after that, the next year again with these bunch of friends, who I'm still in touch with and who are doing their PhDs and postdocs and all across the world right now. I did a project at a place called Indian Institute of Sugarcane Research in a place called Lucknow in North India. It's interesting. I I'm not very keen on I was like not very keen on doing something on plants, but
Yeah.
just happened that way. You have that mob mentality of tagging along with friends and going to places
Of
to
course.
do stuff. But it was very exciting, and we were trying to look at expression of an enzyme called sucrose synthase, which kind of is involved in accumulating sucrose in
Yeah.
different parts of the sugarcane. And I kind of learned about sugarcane for some reason during this project. It was like a 3-month project or so.
Mhm.
Um, and then we were doing like expression, uh, gene expression in different parts of this plant and so on. And that was the first time I really did, I mean, I had done, of course, lab experiments and so on, but like doing a project from start to end kind of thing there. Even then I was not very sure that this is what I wanted to do. And like I said, the whole part where you know in India, probably in the US as well, uh, at the end of your undergrad when you want to decide what to do next, and I'd like there I had appeared for a lot of career placements, and I had got placed in a few companies to do like science allied jobs, and it was like
Mhm.
well paying and it was all good. But somebody I was very confused, and somebody gave me the suggestion that it is always good to be in the area which is of your core competence. And I
Yeah.
always thought science is my core competence. Obviously, I mean, doing some job which is like allied, like I mean, selling some product which has to do,
Yeah. Yeah.
a pharma product,
Yeah.
um, there were curiously, because it was a technology course, an engineering course, we
Mhm.
also did some coding and whatnot. So we could also have gotten placed in like all these IT companies and so on, which is like
Yeah.
completely different from what I learned. So where does
Yeah.
all that go? And I'm glad I did that. I like gave entrances for like the top uh, places for PhDs in India, and I got to go to interviews once I qualified these exams to different places, and like got to meet people, see labs, and read about
Yeah.
people, and that kind of locked me into the whole thing of
Yeah.
actually deciding to do professional science. It was like a short period, I would say. It all happened in 6 months, that yes, I love science, but this is exactly what I want to do. And after that, there was like, I mean, I do not see myself doing anything else now. So
I love
that's how
that.
it is.
It's interesting because the way you were explaining it with so much enthusiasm, I think you've always wanted to be a scientist, but you just couldn't put it into these words. You were just following your interest.
That
Um,
is possibly true. That's a nice
that's
way to look
that's
at it.
that's what I'm hearing. I'm here. So you could have come to the podcast and said, I've always known I wanted to be a scientist, which would have been absolutely true. But I think you were so into it that you didn't have the time to look out and be like, okay, what do I want to do? You were like, oh, I love this. This is what I'm gonna do.
Yeah.
And I think the
Yeah.
advice that you received as well is spot on. I mean, sometimes you have to make a decision and take a job that you don't particularly like because it's the
Hmm.
way to go to wherever you need to go next.
Yeah.
But if you
Yeah.
have the choice, follow your passion as much as possible and do what you like. And also you made a great point around, what be living in, I don't want to say in a safe place where you use your core competencies. I think you need to be able to use your core competencies and be comfortable and not feel like that's a heavy lifting
Oh,
in order for
definitely.
you to learn new things and bring them on.
Yeah.
And I
Yeah.
think that's really, really important.
And I feel like that is what a PhD kind of does to you. I mean, you're you're thrown into the mix where you do not really, you haven't done science at that level really, but you have to start learning from zero from your colleagues, and reading scientific literature, and then you do a new project, and suddenly you realize, and I keep thinking about this when I'm down, that you know the project you're doing is essentially something that nobody in the history of this universe has ever tried to do, or tried to do but not accomplished because it's not in the literature,
Yeah,
right?
having the
So
yeah.
that feeling of being the first to be able to achieve it
Yeah.
is, I mean, it gives me goosebumps right now thinking about
Me too.
everything
I wasn't
that
gonna mention it, but you did, and me too.
yes, yes, yes.
I think and I think it's important as well to put things into perspective. Just because it's not
Mm.
working now, it doesn't mean that it's never gonna work. It's a fine line between, oh, how hard am I gonna push before I say I give up?
Yep.
But I think, at least for me, during the course of my PhD, now that I'm looking back, I feel like during my master's, I learned how to pipette and how to follow protocols. And I
Mm.
have to say, I don't think I was that, that good of a master's student. I just tried to survive the whole thing because I had classes at the same time. And so my time was split between the lab and the classes.
Mm.
And then during my PhD, I feel like I learned to really solidify the skills I gained during my master's. But towards the end, that's when I feel like I matured into the role,
Mm.
because I started integrating what I read and I started asking questions beyond what, for example, my PI was interested in.
Right, right.
And for example, my last paper from the lab, um, the whole my PI came up with the project, or we came up with it together, but the direction it took was completely my choice.
Mm.
And I had to push and say, no, 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 we absolutely have to look for this.
Right.
And the other thing is that I was able to get my colleagues in the lab to help to finish the project faster. And then we all ended up on the paper. And that's something that you learn with time, and that comes with experience.
Yeah, absolutely. And it's you're really right about the environment, the colleagues part. And I was just thinking back, and I think when I was a grad student, like the only thing I was told is that, look, we I mean, at this point, I mean, we are interested in looking at endocytosis and trafficking of this receptor, and that's it. That's the cue. And then from there to like developing, techniques and tools to like do all of that, it was like a free hand to do whatever. And then
Yeah.
with time, you become more and more independent in doing whatever. And
Yeah.
thanks to all my mentors, I mean, both Amit and Manoj, also, I mean, at this point, Manoj is like, he's so, uh, I mean, he gives me all the independence for me to explore whatever I want to do, essentially, uh, because I think it's very important as passionate scientists to be able to explore your own interests, and you just
Yeah.
need to align yourself in some way or the other to the lab you are in as a trainee, I would say. Of course, otherwise it doesn't make any sense because
Yeah.
you're
Yeah.
there for training
You can't be
purposes.
working on zebrafish when everybody's working
Exactly.
on.
That wouldn't make no sense, but yeah, but otherwise,
Unless you can find a link and then convince your boss that it actually makes sense. So it's not impossible,
absolutely.
but yeah.
Yeah. So I'm very happy that, I mean, through my PhD, I think I kind of developed the independence through the course of the PhD. And at this point, I'm so happy that I have a mentor who like gives me the independence to explore essentially whatever I want to do in the purview of, and of course, I'm interested in what the lab does. So that completely makes sense. Yeah.
Yeah, you wouldn't have joined the lab if you were not interested in any of the projects and
Exactly.
output. Um, it's so interesting because you mentioned a little bit about the work that you done during your PhD, and what the lab's focus was, and it was very biophysical, and it was very,
Hmm.
very, kind of structural, and also some computational aspects to it.
Hmm.
And I was thinking to myself about lipids. I mean, we don't think enough about lipids
Oh yeah.
and
Oh
their
yeah.
influence, and it's very difficult to study. But how cool is this still?
I have an entire rant about it.
Okay,
People
please.
don't. Do
This
you
is
want me to go right now?
This is your space if you'd
yeah, I
like
mean,
to.
you're absolutely right. And people, I mean, although we work on a receptor, on rather a large family of receptors, GPCRs, that are embedded in the membrane, but unfortunately, I feel like a lot of people, including a lot of scientists, PIs, just think about the membrane as this slab where there is this
Yeah.
receptor
Yeah.
embedded. And I hate it when people call lipids grease. Oh my God.
I've
My
never heard
blood
anyone say
boils.
that.
Oh my God. And here I am, and I mean, even before somebody who uh kind of studies GPCRs for a living, I would say, um, I kind of identify myself as a membrane biologist because
Okay.
it, I love membranes essentially, and you couldn't have asked for a better system to study membranes than
Yeah.
GPCRs, because they are like 7 transmembrane domain
They're stuck in there.
receptors,
Yeah.
and it's all about the membrane. I mean, irrespective of whether the GPCR is on the plasma membrane or in some other compartment inside the cell, it is always in a membrane milieu. And the fantastic part about membranes is that these are like, I mean, made of diverse variety of lipids, not just compositionally,
Yeah.
but the way they are spatiotemporally organized inside a cell. The plasma membrane is so different from an endosome, which is so different from the Golgi, which is so different from
The
um,
nucleus.
an ER and
Yeah.
or nuclear membrane. I mean, who knows what's happening there? So this is something that people should think more closely about, rather than thinking about it as a slab which sounds so passive and not contributing to anything. So yeah,
No,
it's high
thank
time
you.
to think about lipids.
Thank you. Now, I think it makes a lot, it makes a lot of sense. And it's typically when you listen to talks and people want to get structures or something like that, like they're focused on the way the talk is, I want to say, presented. And I think we're so focused on understanding the GPCR and it's so difficult already that we just don't have the tools to think about what's in the plasma membrane and how does that influence the GPCR function. But we always want to get rid of the fat, and we always want to get the detergent in there and get rid of and get the receptor out. And but then again, I understand that we don't really have the tools
Exactly.
yet.
I mean, I totally get it. And in fact, I mean, the whole point about trying to like reconstitute things is to be able to take it out for, structural purposes, because of course you cannot do anything to
Of
study
course.
the receptor in its native milieu, especially when you're doing like structure. I mean, X-ray
Yeah.
crystallography or cryo-EM or whatnot, right?
Yeah.
But I mean, my problem is not about people trying to reconstitute. I mean, I love reconstitution. The whole point is you take a bottom-up approach and increase the
Yeah.
complexity as and when you
Yeah.
can. Start with like detergent micelles, or now you have nanodiscs, and
Yeah.
or
VLPs even.
integrals,
Yeah.
or GUEs and whatnot. And with uh, improvements in techniques, you increase the complexity, you make it as natural as possible, and hopefully one day we'll be able to study this in the native milieu of a cell, or maybe even the
Yeah.
native tissue and so on.
Yeah.
But my problem is not with the fact that you have to take a bottom approach, because otherwise how can
Yeah.
you, right?
Exactly.
Problem is not being able to identify that this is something that you should even think about.
Okay. Yeah. Yeah.
Yeah,
Not even
yeah.
acknowledging that this is important,
Exactly,
actually.
exactly. That's the thing. Yeah.
Okay, I got you. I got you. Well, it's interesting. I'm not going to mention the name of the company, but it's easy to find. But I was at this GPCR Targeted Drug Discovery Summit earlier this week, and there was this company who actually figured out a way to. They have this scaffold type of thing, and then it actually you get the receptor out of the membrane with its native membrane, and then you can use that as particles. And I thought to myself, how cool is this? Because basically you
It's
could
really
study the
exciting.
receptor in different cell types
Yeah,
and then
yeah,
compare those.
yeah, yeah. And it's very important to acknowledge all of these factors, because now we have a lot of evidence that lipids and the membrane
Yes.
microenvironment does contribute to receptor structure and hence function. You have defined potential hotspots slash interaction sites and even motifs where uh, lipids bind to receptors, or I wouldn't say bind, because I mean, to the pharmacology audience, if you say bind, they would start thinking about nanomolar affinities and so on. And unfortunately, that is not what we're talking about when you talk of lipid protein interaction. But yeah, it is an exciting and an important avenue to consider.
So everyone listening out there, think about the lipids in the membrane and how they influence your receptor function. I think it's very important, yet very difficult to study with in live cells, at least. All right, so the question, what is your favorite GPCR? But maybe I should change it in your case and ask, what is your favorite lipid? Or you have that too.
No, I mean, look, I love GPCRs. There is no doubt about it. Otherwise, why would I be in a Dr. GPCR podcast and like talking to you when I love GPCRs? There is no doubt about it. But as it turns out, I really don't have a favorite GPCR per se, but there are GPCRs that are close to my heart, and it turns out that those are the ones that I've worked with. I mean, during my PhD, I worked on the serotonin 1A receptor. It's a class A GPCR. Curiously enough, it was the first GPCR ever to be cloned. So it's really cool that way. Yeah, and I worked on that receptor. Now I work with a bunch of receptors. I work with the beta 2, of course. How can you not work with beta 2 if you're
like the
doing
classic
anything
one.
basic with GPCRs? I study the delta opioid receptor. And different aspects of it. So I like always like to step on a couple of boats to kind of mix and match projects and try to get as much from different places, because it's fun, because it always happens that something or the other doesn't work at one point in time. So you have something else that you can fall onto, I guess. So in the lab right now, I do high resolution total internal reflection fluorescence microscopy to look at organization of receptors in clathrin coated pits and how the membrane microenvironment kind of has a role to play, or how the cargo actually influences the membrane microenvironment. It's both ways. And then I'm also looking at trafficking of delta opioid receptors, which is a very interesting and very translatable aspect, and something that Manoj's lab has been studying for quite some time now. Yeah. So
that's really
I would
cool.
say these are the kind of receptors that I kind of like. As far as the lipids go, I would say, because I've worked mostly with cholesterol till now, that it's a fantastic lipid. It's an amazing lipid, I would say.
I was sure you were gonna say cholesterol. I think, but I don't know why.
I mean, if anybody goes through my publications, they would know that I love cholesterol.
It's cholesterol.
It's cholesterol. And cholesterol is a very exciting lipid. It's not a phospholipid. It has a small head group and like these sterol rings, and it's completely different from any other lipid. And it changes, rather it influences membrane properties in more ways than people can imagine. So it's an exciting lipid.
I love it. I love it. Speaking of lipids, and this is kind of an open question. What do you think, what tools do we need to better understand the function of the role of lipids in receptor function?
Yeah, so that's an interesting question, and something that people have been really toiling with for quite some time now. So I look at it from two perspectives. When you say the function of lipids, you can think of lipids as an individual entity. For example, cholesterol is one lipid, or sphingomyelin is one lipid, or phosphatidylethanolamine is one lipid, and so on. So individual lipid species. And the other is trying to look at the lipids as a bulk, as a milieu that creates the membrane. And then you can talk about the properties that this combination of lipids imparts to the membrane. So as far as the techniques go, there are these two aspects to it. I think the biggest challenge when you're looking at individual lipid species is the transience of these interactions, and especially in a live cell environment, to be able to capture that the transience of the interaction and being able to say that, okay, yes, this interaction makes sense within all that noise that surrounds it, uh, is a tough question. So now we have, as far as protein interactions go, these like solid tools like BioID, TurboID, and then Apex and whatnot, right? That capture these interactions around the vicinity of a protein of interest. And a colleague of mine in Manoj's lab, he always keeps talking about um, an apex for lipids and something like that, something that can capture these interactions in the spatiotemporal scales that are relevant to these interactions. I mean, you could capture an interaction, somehow artificially induce it maybe, but being able to like do it in the scale in which it makes sense is a challenge. As far as properties of the membrane go, I mean, people have and are developing probes that can report on various membrane physical properties. You can talk about how ordered or disordered the membrane is, or what is the polarity of the membrane, or how thick or thin the membrane is, which is causes hydrophobic mismatch and can cause tilting of your transmembrane helices one way or the other. And we know that these are important aspects in rendering signaling or non-signaling conformations of receptors. uh, we have probes, but being able to kind of utilize these probes to do imaging in live cells, such that you have enough space in the spectrum to image multiple things at the same time, is a challenge. And we have things coming up, and we need to utilize them more judiciously and come up with experiments that can really give you some interesting data.
No, it's interesting, and I think I was talking to Anthony Boucard, who works on adhesion GPCRs, and I think this question that I just asked you, or this train of thought, was asked in the form of, as if you had a magic wand and you could come up with any technology or to better understand adhesion GPCRs. I think his answer was like, I would make myself so tiny and go visit the adhesion GPCR. I think in your case it would be go visit them, go observe directly the lipids and the different types of lipids and how they interact, and how they not necessarily interact, but have influence.
Sit inside the GPCR and look at the lipids, and sit in the membrane and look at the GPCR, both ways. So essentially what I'm trying to say is we need probes on both ends, and which can give us like good data.
And I think we're getting there slowly but surely. I also think that we don't know enough yet to link maybe receptor function to a disease and how lipids influence, or can we use the lipid environment to target the receptor in specific tissues versus others. So there's a bunch of work to be done.
Absolutely.
I will still ask the question, although I know the answer is yes, and 100% of the time the
GPCRs are drug targets. I
know that they question. Are.
I know the answer.
I think they're great drug targets. I think there's a lot of work that was done that still needs to be done to better understand them, but also to target them. I think we've gotten really good at understanding how GPCRs function. For some, we've linked function or dysfunction to a specific disease setting. We were able to come up with antibodies, small molecules in that context to modulate receptor function. One of the difficulties I see is, okay, it works in a cell, hopefully it works in an animal model, but then it fails when it ends up in humans, if it at all ends up in humans.
Absolutely.
Anything
and that has something to say about the systems as well, right? I mean, so much of the structural information which feeds into a lot of the drug discovery aspect of it comes from these reconstituted, purified systems, which are not really native. But how can we know about the native systems and translate the whole thing, like you said, is the challenge. And yeah,
Yeah.
it's exciting times, I would say.
I think so too. I mean, this conference that I went to, it was just such it was a nice balance between academic and private sector presentations. And there was a lot of really, really, really cool data shown. I mean, I got out of there like, I'm exhausted, but this was great. It's like, I feel like I was at a rave where we were all talking about only GPCRs. Not that I've ever been to a real rave, but I think this 3 days were like
Yeah,
really, like I was able to eat, breathe, see, and get involved and learn more about what the field is doing on both ends, basically on both spectrums.
sounds so
Um,
exciting. Yeah.
yeah, super exciting, and we've talked about it before we hit record. But if you don't mind, I'm gonna bring it up here as well. We're talking about being able, when you were interviewing for a
Mm.
PhD position, you interviewed at different places, you were able to meet different scientists, and it helped you also gauge your interest. It's hard
Yep.
to know what you're interested in unless you started doing it. And I think one nice way to do that is to go to conferences.
Yep.
And I know you guys are still in the organization phase, but if you can touch upon the next meeting that you're involved in, that you're involved in from an organizer's perspective, so that we can pique people's interest
Yes.
and then ask them to mark their calendar for a date. I'll let
Yeah.
you.
Yeah. So watch out for the third Transatlantic Early Career Investigator ECI GPCR Symposium, tentatively scheduled for September of 2023. I'm sure a lot of the listeners of the podcast might have heard or even participated, I would say, in
Yeah.
the first two editions, which were a huge success. And these
Yeah.
are trainee focused uh, meetings, which give trainees the platform to convey their science. There is no registration fee. You do not have to travel. I mean, it could be an unfortunate part that you don't have to travel, but there is no registration fee. You don't need to really ask your mentors or bosses for funding to travel. You just, all you need to do is sit on your computer, make sure you register for the conference, and have some cool data to present, either in the form of a talk, which we would select from a bunch of abstracts that we will seek soon, um, or on a poster, and like meet your contemporaries, trainees. Graduate students can meet fellow graduate students, can meet postdocs, ask about potential labs that you want to be in the future, make contacts. We will have a bunch of um, you know uh, very famous scientists from the GPCR field. You can look up the websites for the first two editions, and you will know who they are. They are the creme de la creme of,
Yes,
I
I
would
was
say,
thinking the same thing. It's funny
of the
because you and I
GPCR
have
community. Sorry.
haven't met in person and haven't well, we've interacted very little, and you took the words out of my mouth. I was thinking crème de la
Yes.
crème, so
Although not with such a nice French accent
French
as you
accent,
do, but
yeah.
yes, I try. So it's a fantastic opportunity to meet these people over networking sessions and lunch sessions, all from the comfort of your desk in your lab or at your home.
Yeah.
Based on the time differences, of course, we try to uh, work this out such that it like is in a reasonable time frame for at least Europe and the US, which span a whole, you know,
Yeah.
range of time
Yeah.
zones, but we
Yeah.
try. It's a great opportunity to network, to convey your science to an audience that would appreciate it, because it's mostly going to be trainees,
Yeah.
to get feedback, to get recognition as well. Yeah, and just feel a part of the community without the hustle of trying to,
Traveling.
travel or find money and so on. So
Yeah.
look out for an announcement soon on Twitter, or we'll have emails sent out, and
Yeah.
yeah.
Yeah, and as soon as you guys have a website, we're always interested in collecting information about upcoming events. So in the ecosystem, there's the events section. If you're looking at what next conference you should be going to, that's the place to go. I think this is the only event that is dedicated to trainees.
Yes, absolutely. And that's the selling point of this meeting. It is for the trainees and by the trainees.
Yes,
So,
exactly,
so
exactly.
yes, you need to be there and be a proud trainee because
Yes.
someday will not be, unfortunately, or
Eh,
fortunately.
Well, yeah, I mean, when you think about it, every time you go to a conference and you see a PI present, you're like, oh my God, how great work he or she has done. But unfortunately, I don't know, I'm trying to come up with I'm drinking tea because I've had one coffee already today. But what I want to say is that it's not the PI who was pipetting. It's all the trainees, and unfortunately, I don't think trainees are given enough credit in general.
Yeah.
And they do deserve the credit because the trainees are the ones who are pipetting, the trainees who are the ones who are really dedicating hard work and time to do the research.
Yeah.
I'm not
Yeah.
saying that PIs are not supportive, but there's, it's now that I don't work in the lab anymore, it's so easy to say, oh, do it again. Have you missed this control?
Yes.
But then I realized the effort that it takes to run these assays and to develop them and make sure that they're robust. So I think it's a great opportunity to have this day dedicated to trainees.
Yes, absolutely. And I'm looking forward to, we are all actually looking forward to participation from many of you and to take this, actually to take the tradition forward, I would say. And that's the most important thing. And, you know, there are so many initiatives that begin, but unfortunately do not
Yes,
sustain. This is
yes,
something that we in the GPCR community want to keep alive and take forward. And our predecessors have done a fantastic job
yes.
at starting. And I mean, what a start. I mean,
Yeah.
it's almost like the bar is so high now that we shudder to think what we would do. But yeah, it's a
Yeah,
great meeting, and you get to
yeah,
meet such fantastic people doing such
yeah,
awesome science.
yeah.
And these are all your contemporaries, so it's fantastic.
Exactly, exactly. And I've interacted with all the organizers so far. The one thing that I could not get them to do is to come on the podcast and talk about the behind the scenes of this meeting. And I think because when until you do it, you don't realize how much work it takes.
Hmm.
How it's a process development, it's a logistical nightmare if you
Hmm.
haven't done it, and you gain tremendous experience by doing these kinds, making these events happen.
Yeah.
Which gets me to the next section of our conversation of the podcast is, I always ask the question about advice to young scientists who want to contribute to the field. And
Hmm.
before you answer, I want to make a point, or I'm going to try to make a point, is
Mhm.
that when you're a trainee and you come out of a lab, you do a postdoc or a PhD, and you go to get what quote unquote is a real job, you think, or at least it was my case, and let's talk about me, or at least take me as as the example. Hopefully I was the only one, but I always thought, oh, I only know this much from a
Hmm.
scientific perspective. And I never thought about what they call soft skills.
Hmm.
And you develop those by interacting with your colleagues. You develop those when you organize meetings like the one you guys are putting together, because then you still using science, but you use your organizational skills, you use your communication skills. And yes, it's a nightmare to even think about doing something that you've never done before, but once you've done it once, you have proven that you can start something from scratch.
Yeah. Yeah.
And I think that's very
Absolutely.
valuable.
Absolutely. Absolutely. And it teaches you so much. Science is a community affair. I mean, nobody can do science in isolation, sitting in a
Yeah,
lab pipetting. I mean,
yeah,
least of all, you would need suppliers to give you the chemicals you work with. So
yeah,
it's a community affair, and it gives you a sense of the community, and you're giving back to it when you're
Yeah.
actually organizing a meeting, because
Yeah.
yeah,
No, I
it's
agree.
a part of community building and
I
feeling
think
a
so
part
too.
of the community as well. Yeah.
Exactly, exactly. Which is, and on that point, this is Dr. GPCR's mission is to build out this community.
Yep.
I think we've gotten to a point where most people in the field know about Dr. GPCR. So it's funny because I go to meetings and people look at my name and they're like, "Pff," and then under it they see Dr. GPCR and they're like, "Oh, it's you." And
You are
I'm
the biggest
always
celebrity in the GPCR field right
I'm
now.
so humbled. But the whole thing started with the idea that we would like to have a community, a place where you can go and complain that this experiment and these cells didn't work. Anybody can have any idea, what can I do? But a place where we can celebrate our successes together. Because there's one other thing in science is that it's a lot of failures. And I always say it's a dopamine rush to a dopamine rush, but then the distance between the two dopamine rushes varies. Sometimes it can take years.
Mhm.
And whenever we have successes, we have to celebrate them.
Absolutely.
All right.
Absolutely.
Advice for young scientists who aspire to contribute to the field.
Well, you almost spoke about one of them. So let me start from that. I really feel that um, the most important facet that one should possess as a scientist is patience, I would say. Like, be patient, and just keep at it.
Yeah.
Science will reward you, and you will be rewarded for your work. Just be patient, keep at it. Also important at this point also to know when to, like, stop being keeping at it, because you don't
Yeah.
want to keep at doing bad experiments, because you discover at some point that there was something wrong about it. This just doesn't work. And
Yeah.
as trainees, it's also important to be aware of time frames, because nobody wants to be a trainee
Yeah.
for their whole life. So yes, all these practical aspects are true, but I feel like the most important facet, and I'm saying this upfront because you touched upon this just before I could
Yeah.
answer,
Yeah.
is to be patient and believe that dopamine rush is around the corner, and
Yeah.
it will be there. And when you reach that point, you would just want to go from that one to the next one, and that's what keeps us going as scientists, I believe.
Yeah.
So that's point number one, I would say. Um, Apart from that, I feel like it's very important to surround yourself with um, positive people, uh, and people who you admire, not necessarily only as an intellectual or as a scientist, but also as a person, because ultimately I feel like we are all, of course, we are all humans doing science, and it's a very personal opinion, but I'd say you have to be a good person to be a good scientist, and uh,
Yeah.
that will, like, keep you going. Otherwise, there's a point if you, like, just be a scientist and not work on yourself. Someday it will cave in, and that might cause
Yeah.
some trouble. So be positive. Surround yourself with positive people. There is a lot of negativity around us, but surround yourself with positive people, and I would say, yeah, try to be a better human. And if you're passionate enough, you'll always try to be a better scientist, because there is nobody called a best scientist.
I don't think so either. I think it's sometimes, well, it's always hard work. Sometimes it's dumb luck.
Yeah.
And I think most of the time is you going to conferences, for example, and getting to meet people who say, "Oh, I'd love to work with you on that."
Mm. Mm.
And I think, at least in my case as a trainee, and it took me a while to understand this, is that at the end of the day, you don't have to be an expert at everything. You cannot be an expert at everything.
Absolutely.
And if you want to, advance science, you don't have to do all the experiments.
Mm.
You just need to figure out what are the experiments that will lead you to answer your question. And if you don't, and also think about the time frame that you have, the timeline, and go find the person who's really good at it.
Yeah.
To your point, is in their comfort zone, different
Yeah.
than yours, and then can add to that work.
Absolutely. Absolutely. In fact, I mean, I I almost feel like the part that is more exciting being a scientist is thinking about the experiments rather than doing the
It-
experiment. It's almost a PI mentality, and I want to be a PI someday, hopefully soon. Uh, uh, but yeah, it's just the whole part about thinking about science every day you go to sleep, think about this experiment or that experiment and how you could have done that well, how you could like plot that data in a more presentable manner,
yeah,
or stuff like that.
yeah.
That's the most exciting part of it, I say.
Yeah. Yeah, but I have to also add that I feel like I had the best ideas when I was not in the lab and I wasn't doing experiments. So I think
For
there's a
me, it's
need.
mostly when I'm taking a bath, I'm in the shower. Yeah, that's
Well,
when most exciting ideas hit me.
that's when I typically start, my brain starts working, and then I come out and then realize I forgot to wash my hair and I have to go back because my brain is focused on, okay, what do I do? How do I do things? But I think, I like to walk, for example. I used to have a dog, so my dog and I would walk, and like he'd be doing his business, sniffing stuff, and I'd be like, okay, maybe I should do this, I should try that. And I think there is an important aspect of relaxation. Let your brain, you know, your brain work on the problem while you relax, because
Hmm.
when you get back to the lab, that's you might get an idea that you didn't have before. And I think the other aspect is really having discussions with people who challenge you.
Definitely.
Surround yourself with people who are smarter than you, who keep asking you, yes, but why?
Yes.
But why? To the point where you, at some point, you say, I don't know.
Yeah.
And it's a difficult exercise, but
Hmm.
it's very good, because that's when you realize that,
Hmm.
oh, there are flaws, or there might be flaws, or there's ways to improve on your thinking, on your experiment, on your research.
Hmm. Yeah, yeah. It's I think, and there is this article in the Journal of Cell Science. I think that it's called "The Importance of Stupidity in Science." I think that's what it's called. It's really, I think that is the most important advice that I could have received as a trainee, because more often than not, it's important to feel dumb as a
Yeah.
scientist, I would say.
Yeah.
Uh, Otherwise, there is no scope for growth. If you feel like, oh, yeah, I've achieved it, I know it all,
Exactly.
then you're not really a scientist, I would say. So yeah,
Exactly.
it's important to feel stupid and not be worried about it.
Well, it's difficult, it's very difficult. I was
Yes.
sitting at this conference, and there was this presenter, and it was pure molecular pharmacology, like, Terry Kinakin
Mm,
style
mm.
data presentation. And I felt this big. I was like, I don't understand any of this.
Yeah.
But
Yeah.
then I had to put into perspective that that's why I was sitting there
Yeah.
listening to it, so I can learn. And the other thing is that I have strengths that other people might not have,
Exactly.
and use your strengths and get comfortable, and then you take that opportunity to learn something new, and then increase that portfolio of strengths that you have. Never
Yeah.
ever ever stop learning.
Yeah. And I would say, I mean, platforms like these, like the Dr. GPCR podcast, I'd say, I've what it's been, at least the ones that are published as of now are more than 100 recently, I know.
Yes, 100.
And through all of these and many other podcasts or like seminars that I listen to and so on, you just keep listening to so many stories and advices about how people have uh, gone about their business of being a scientist, and you learn from it. And it's important to not shut your ears and be aware of what people are doing and how
Yeah,
they've achieved what they've achieved, and also excites you. I think the best part is like going to conferences when you just the whole aspect of listening to exciting science. Uh, it gives you more impetus to like go back to the lab and do that one extra
yeah,
experiment that you
yeah,
know
yeah,
that you were kind of postponing because you thought, I don't know whether this would work or not. But then
yeah.
that's it. Just the excitement, the like the dopamine that you say,
Yeah. We're
or the
all, I think
adrenaline,
we're all
whatever.
junkies in that aspect, as from a scientific perspective. We just want the experiments to work. We want to come up with ideas. We want to test them. And when it works,
Yeah.
dopamine. And then you're like, this is great. I want to do more. I want more.
Yes. Yes.
All right. Last but not least, last question. Top three aha moments that changed your trajectory, that influenced your trajectory as a scientist.
Yeah. I mean, one is the realization that I want to be a scientist, I would say, and I've kind of answered all of these questions in some form or the other. But actually, the realization that I want to be a scientist, this is what I want to do when I was like interviewing and going
Mhm.
to places and just soaking in the atmosphere. I mean, when you go to a lab and when you go to a, I don't know, university campus or a research institute, whatever the form it may be, there's something different about the air there. And you see the
Yeah.
excitement in people, and there's something interesting going on in one of those test tubes or in the incubators and under the microscopes and so on. And that whole atmosphere kind of, I felt like, oh yes, aha, this is the thing that I want to do. And
Yeah.
that was,
Yeah.
I think, a very defining phase, I would say, not a moment. I mean, I would always, I think most of my answers would be in terms of phases, but not moments, because
Yeah. Yeah. Of course. No, no.
yeah.
I think, you know, sometimes it's just realizing that something has not been working and you're like, oh, and I finally figured it out. That's also
And
an aha moment.
that's my next one, actually. And I'm glad you kind of hinted at that. In fact, I remember so after I started like working, and the real big aha moment was when I actually acquired my first confocal image. I
Mhm.
remember for months, and I felt so dumb. I was just trying to label actin in cells, and I just wasn't able to for some reason. And people would say that you just throw this label into cells and it'll label. What is wrong with you? Why can't you do this? And I was trying so hard, so hard. And one day, finally, um, turns out that the bottle of formaldehyde that I was using was bad. And then by sheer luck, I ended up using some other bottle and it worked. And I remember like peeking into the eyepiece of the confocal microscope and looking at the cells with such bright red fluorescence. I cried literally. Oh
Look,
my God. It's as simple as just labeling actin, and people do it
all the
every
time.
day.
Yeah.
Yeah, right. And but the feeling of looking at it and like seeing that bright red fluorescence,
Yeah.
it was like, oh my God, this is what I'm here for, and I want to do this. So,
I
yeah.
love it. And by the way, I want to mention to the audience, we did not rehearse this at all.
Oh,
So,
that is so.
because you mentioned I hinted at it at your second aha moment, but
Yeah.
it was just sheer dumb luck, basically.
No, I know. I mean, we are all scientists, right? So
Yeah,
we
yeah.
know,
So
we
maybe
know
I
what works.
had a
Yes, we
vibe there.
think similarly. Yes. Yeah. Yeah. So yeah, and I would say, I mean, whenever I've like gotten a result that is completely um, shocking in a sense,
Mhm.
and I mean the first um, reaction would be, oh wow, that's so exciting. And soon after, immediately, because you're a scientist, you think, oh my God, I've done this only once. What if this does not work again? And you repeat it and you see the same thing happening again and again. And then
Yeah,
oh my God, this
it's
is so
good.
exciting.
Yeah.
So I think there have been a few of such aha moments in uh, my shortish career as a scientist till now. So, and yeah, that's what like keeps you going. The first time when I saw that, when you like deplete cholesterol in cells and you see a complete switch in the trafficking uh, regime of the receptor that I was working with, I was like, oh my God, that is so crazy. Why should that happen? And it happened always. It happened every time I did that experiment. And now whenever I do an experiment and I see some result that repeats itself, and you never expected that to happen,
Yeah.
that gives you a kick. Yeah.
Yeah. No, I agree. I agree. And it made me think of the last project I did during my PhD with CCR2,
Mm.
and the whole project, I'm not gonna say it was easy, but it was smooth.
Yeah.
I think all the figures were an n equal 3, and
Mhm.
I did them 3 times in a row, basically no setup, because that receptor behaved amazingly.
Yeah.
And I was like, wow, this is really cool. And I think this is the one paper where when we sent it for review, and it's a molecular pharmacology one, and there were no change, no comment other than this is a great paper, amazing. There
Nice.
was one reviewer who said something around at the bottom of a table there was ND, and they wanted to know if it's not determined or not detectable.
Oh,
And that
wow.
was it.
That's
That was
amazing.
it. And I was like, wow, this is cool. On the other hand, I worked on a different receptor, CXCR3, which is Sudhir Rajagopal's favorite
Right.
receptor there. And that was such a challenge because I was working with the splice variants,
Hmm.
and they were especially the cyclic AMP experiments. I think I repeated them 10 times because only one out of the three would respond. And I thought maybe I did something wrong. Maybe it's the chemo. What happened there?
Yeah. And, and
It was
and
that was painful, but it was
yeah,
fun.
yeah. And nobody should get me wrong. I mean, to reach these aha moments, you have so many oh no moments in the
Yes.
way that that aha really sounds very satisfying when you reach there. So
Yes, exactly. Maybe we should incorporate like top three oh no and top three followed by top
yes,
three aha moments.
yes, yeah.
Amazing, Aditya. Thank you so much for being here today. I really
Thank you
enjoyed
so
talking
much.
to you.
I thoroughly enjoyed this. Thank you so much for having me. I mean, and I'm looking forward to so many more episodes of the podcast.
Thank you. And I will be there for that for the event as soon as you guys have a date. I think
Sure,
I have some extra free days. If not, I can just tune in and listen to the exciting talks from trainees. All
definitely.
right. I'll talk to you soon. Don't go anywhere. Thank you for listening to this Dr. GPCR podcast episode. We would like to thank our guest, our Dr. GPCR team members, Attila, Ines, Monse, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with us and with our partners directly in the ecosystem, so make sure that you join us today. Also, please subscribe to the Dr. GPCR newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial on our website. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. You can always email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.